Hi, this is Shuman.
Hi, Shuman.
I am Benita.
Hi, Benita.
We have Denis with us today. Denis was born in Geneva. He earned his PhD in biology in 1984 and is currently professor of developmental genetics and genomics at the EPFL and at the Department of Genetics and Evolution of the University of Geneva.
Denis has long-standing interest in the function and regulation of Hox genes. His work in Hox genes has a tremendous contribution in understanding embryonic development. He is a fellow of Royal Society and a member of National Academy of Sciences. He also occasionally writes columns in Science and Environment. We welcome you with us today, Denis.
First, I'd like to thank you guys. It's a great honor for me to come to the kitchen lab. I love the idea, and I'm very glad actually you welcome me. For a very long time, we are interested to understand the regulation of gene activity during development. This is what I'm interested in. How do genes are regulated to be expressed in the right cell at the right time, such that an embryo can develop properly and give a human being or a mouse or whatever. And to do this, we have used a particular gene family, which you mentioned, Benita, which are the Hox genes. And for some reasons, which are not so interesting actually, but for some reasons, they are extremely prone to these studies. And this is why my laboratory has spent essentially 35 years trying to understand how these genes are regulated. What is the signal that activate them? What is the signal that says, now you stop? And all of this, that's the last thing I want to say, all of this in an evolutionary context. The work is a work in developmental biology, molecular biology, but the thinking, the context is always, how did that happen? How did it start? How does it work in other animals? Is it the same? Where do the genes come from? That's what I'm fascinated with. And now I'm ready for the questionnaire.
Our first question is, when did you fall in love with science? Any particular incident or the time you remember when you decided that, oh, I love it and I want to do it?
No, I actually never did. I've never been in love with science. I've always been in love with nature, always loved animals. I always loved trees, flowers, mushrooms. Ever since I was very young, in the countryside of Switzerland, I was trying to catch snakes and things like this. And I could have become, a vet or someone working in a botanic garden, whatever. I think I would have been very happy. It happened that I ended up doing research, and I ended up trying to see what is inside animals, mechanism and so on. But that never really took me off my love of animals and nature. I can look within a mouse in the morning, and I can be fascinated to see a fox in my garden in going back home at night. I love it. I love animals.
I think that's a fair way to fall in love with nature and do science alongside. Since you were interested in looking at animals and observing animals, did Hox genes strike from there that how?
No. My first love was botanic, mushrooms, trees, flowers, and so on. So I registered to make a PhD in botanic, in fact. And because my best friends went into a different place, and I went to visit him, and I found these sort of things, totally random things happening here and there. And then I suddenly found a very interesting PhD place to work with mice. And then I started to work with mice. And then, 4 years later, I ended up in a place, and someone asked me, it was just in the beginning of the Hox story, even before it was published, before the paper by Bill McGuinness and Walter Gehring was published. Yeah. The person had seen the manuscript. It was Pierre Chambon in Strasbourg. And he asked me, do you want to work with this? Because I have the feeling it's going to be a fantastic story. And I said, yes, of course. But, I said, yes, I could have said no. It was this sort of a turning point, these decisions you take more by feeling, more by sort of trying to feel what is going to happen, rather than saying, look, on the plus column, on the minus column, there's pa. And as a result, I've decided to do this now. For me, it never happened like this.
Pretty exciting, and like that is pretty cool to like go with the flow and decide whatever happens, we'll see.
We are excited to know how a day in your life looks like and how you manage to find a balance between your work and life.
I never did. You know, because for me, there's never been any separation between life and work. Life is work, and work is part of my life.
I see.
And my wife is the same, has also been working very hard, and the children were there. And I'm never asking myself the question. Whenever I was not doing science, I was thinking about science. And whenever I was not doing with my family, I was thinking of my family. And I've never seen the okay. Now, you know, the question is, if you have a passion, for example, if you like music, are people going to ask you how do you make the separation between music and your life?
No, of course not.
No, it's no. So, you know, and I think I would even say the contrary. I think it is very important to admit this at some point of your career, and not to feel guilty of spending so much time in the lab. Okay, saying perhaps I see my children a little bit less, but when I see them, it's, you know, quality time. And also, I don't think it is very frustrating for children to systematically hear, you know, be careful, mom is working, you know, she's going to come back late, or dad is going to come back late. I don't think it's so bad, you know. I think it gives them a sort of a way to think about the reality, which is probably pretty good.
Yeah,
I think that's the best definition I'd ever heard, like the way you put it with the music thing. Like, we never separate music from the work or from the life. So why to separate the work and the life differently? So you have been doing science since a very long time, like since a few decades now. Which was the most challenging phase of your career?
Right now.
How?
I tell you that I never felt I was in a, you know, perhaps because I always felt so lucky to be able to do, you know, my passion, to be paid for this, which is completely, you know, unrealistic, in fact. I never felt really, but now it's different. The first thing is that the science goes so fast and the technology evolves so rapidly that you know, for the past years, I no longer had the capacity, the time to cope with this technological development. And in the lab, it's not so good, because I can still talk to my colleagues, of course, but I talk to sort of, you know, a theoretical level and so on. And I can no longer really impact on the precise. And, you know, as soon as I understand how a new technology works, it's obsolete. There's a new one that has came, and then you know, and so this is difficult. And the second thing is that in Europe, mostly in all countries in Europe, when you reach a certain age, you're asked to, you know, stand up and leave your place and go for retirement. It's an obligation. It's not like in the States. So I'm not exactly yet at this time, but I think I need to, you know, at some point you need to start thinking about it and try to organize a phasing out that is as successful as you're phasing in. And so this is a challenge. Yeah.
absolutely.
Do you think that taking retirement is a challenge?
I think it's a challenge for the person who retires, because when you know, but sometimes it's like asking someone who is in love with music to stop listening to music, and it's very frustrating. And it's also a challenge for the structure that you live, you know, for the people who are with you up to the end of your lab and so on. So you need to change a little bit the way you implement your research. Yeah. In the United States, you would stay because you would bring funding, you would bring money to the institution.
Yeah.
In Europe, many people try to stay, but just by using the funding of the institution. And
Yes.
of course, if you do this, it can be extremely interesting for the institution to have experienced people and so on. But yeah, there are young people waiting for this funding, waiting to start with energy, with totally bold ideas, and I think the balance is clear, is clear. So there should be something, a new structure, thought and organized to allow people to stay, but not to prevent young people to
enter in. Yeah, because the number of scientific institution are like growing in a slower pace than the number of students who are doing PhDs and postdocs
yeah,
and trying to become PI or like doing such.
absolutely. I think if I would be able to improve academia, I would certainly go backwards in the start of independency, trying to give independency a little bit earlier to crazy ideas, okay, and then having a subsequent selection later. But you cannot do this if you don't make space and funding free.
Coming to our next question, we would like to hear the Eureka moment of your discovery on Hawk's gene.
Yeah, I went to, for some circumstances, I went to a postdoc in a medical faculty in France, in Strasbourg. And after a few months, the person who was the head of the institute heard about this work on Hox genes.
Mhm.
It was in 1984.
Okay.
And then he asked me if I was interested to take it over and try to go for it in the mouse, because that at that time it was in the fly.
Right.
And he told me, if you agree to do this, I give you a small lab. I give you a space and a small lab for you. And that was barely 6 months after I had joined the laboratory.
Right.
And I said, of course, but I had no idea. You need a bit of naivety, to do this. You don't need to sit and think, am I able to do this? Can I do this? What it means? No, you just go, and it works. It works. It doesn't work. It doesn't work. And but then it was so, the whole field was so dynamic at that stage. There was such a momentum and a lot of interesting things, which I'm not saying it doesn't happen today. I think it's the same today, actually. But it was so dynamic that it worked, and then I never thought of changing. It was so interesting, and then we went on and went on and on, and then I changed institute, and I kept on going and going, and then suddenly you are 65 and you need to start for your retirement. That's how it works, and no, it's a quick journey.
Time flies when you are
So
doing
fast.
something that you love doing so much, right?
Absolutely. You go to, you go to a concert, you don't like the music, it sounds so long, and you go to the opera, 5 hours of opera, it's so nice, and it goes like this, and
Right.
it's very relative. But once again, I think I never took a formal decision of myself saying, what am I going to do next? It just went on and on and on, because I was aspirated by the quality of the problem. In fact, when you try to hit something that is so interesting, you just get aspirated, and
That's great.
I cannot stop myself from asking this. What do you see the future of Hawks is?
Oh, as the future of everything, I'm a complete optimistic scientist. I'm not a complete optimistic human being when I see what is happening everywhere and so on. So I'm rather pessimistic on our capacity to continue doing what we've been doing. But if you can continue on the same pace, it's extraordinary. I mean, the tools that now you can implement are so fantastic. I mean, going to single cell, single molecules, to see things going there and how cells are moving right or left. And, it's extraordinary. Hox genes are not particularly outstanding on this. You know, there's a corpus of what we call epistemic system, that is systems that you have built with results, but also with concepts. Okay, and if you cultivate them well, like like a tomato tree, if you cultivate them well, the system gives you a lot. And you should not go too harsh with the system. You know, you have to respect your epistemic system. You go step by step, and then you get a lot. You know, it's like a sort of a pool of stem cells. You know, you have to cultivate them gently, and then you get a lot. If you go too harsh, then it goes and it
take this thing down.
Yes. So I'm sure that the future generation will have a quality of problem and which is extraordinary, and the capacity to solve them, which is a lot of pressure on you, because of course, when you have the good questions and you have the good tools, what is left? Hey, the only thing that is left is your capacity to do the right stuff. Okay, this is a pressure. 40 years ago, we did not have this pressure. We would say, oh, I've done this because I couldn't do anything else. This I can't do. So I've done this experiment. Okay. Yeah. Today it's more complicated today, because today you can do, so many things.
So everybody goes through experimental failures, and there are few experiments that keep failing for a very long time. So what was the major experimental failures that you had, or how did you manage to overcome it?
I can't remember going back home saying, oh my God, it's failing again. The one I remember is that we tried for a long time to produce mitotic clones in the mouse, because it would have been extraordinary. There was, for many years, people were doing in flies, and it was so interesting what they had produced by doing mitotic clones in the embryo, in the larvae, and so on. And we tried, and of course it didn't work, because the invertebrates, the chromosomes are not pairing in mitosis. Okay, in flies they pair, so that you can induce crossing, mitotic cross. So, but because it failed for about 1 year, or almost 1.5 years, 2 years, then we decided to go into meiosis to make recombination and to make clones. And it worked. And because it worked, we could design a new tool in genetics, which allowed us for 10 years to build chromosomes at a time where the whether the CRISPR would not exist, of course. So it was very complicated, and we could do this because of this failure. Okay, if we would have been able to produce mitotic clones, perhaps we would have produced 1 or 2 boring papers on mitotic clones, and we would not have had the idea to develop the meiotic clones. Okay, so the notion of failure is very relative in science, and people should never consider a failure as a failure. A failure is just an experimental step. Then you have to decide what to do out of this failure. And, it's so often it is as important and as interesting as what we call a success, that is a result.
So what are the practices and what were the philosophies that you followed through your PhD and further career that helped you make the best out of yourself?
Well, look, first, I don't know if I ever reached the best of myself.
Okay.
if I would tell you I reached the best of myself, it would be both presumptuous and very depressing, in fact. So again, I think the only good practice, there are 2 practices. There's your position towards science, and there is your position towards your colleagues. These are 2 different things, but in fact, it's one thing. And I think the correct practice is never to be in a posture, that is to always be what you are. Whenever you make science, whenever you talk to people, and it took me a long time to realize this, not to implement, because I'm not sure I can implement this. Okay, it's complicated, but at least to realize that this is what people should do. You need to keep standards. You need to keep honesty, to evaluate the results for what they are. And you need to do the same with people, to be honest, frank. Sometimes, sometimes it can be a bit tough. So I would say this is the only process that I would think of. That is, science is a human activity, and you need to do it as a human being. And it doesn't call for any particular qualities. You just need to be what you are. And if you can manage to do this, I think things are cool, because you gain the respect of people, because you're just a normal people, and you also gain probably the respect of yourself, because when you go back home, you don't ask yourself, what the hell did I do this? It wasn't really, the stuff I should have done, and so on. So, I think that's the only process that has to be respected, in fact.
So social media is playing a very big role in popularizing science these days. And what are your views on impact of social media in today's science?
Well, first, I should mention I'm only on Twitter. I'm not on any other social medias, and I have a mitigated. I think it's great. For me, it's great in terms of information. I would say more than half of the publication I see, I read. I read not in details, but they are coming from Twitter. So it became for me one of the main sources of information with my colleagues and so on. Okay, but on the other hand, it's very short, and people should really realize that life is not Twitter. Science is not Twitter. Science cannot be, a publication cannot be summarized in 140 words. Okay, and one should be very careful with this, because when you try doing this, you often are a little bit outside of the truth. Okay, so of course this is Twitter. So people going on Twitter, they know that they are on Twitter. So there's all sort of a vocabulary, and to expose yourself to which I think is much easier for your generation than for my generation. But I can see that there are people from my generation who behave like a Twitter freak. So I think it's okay. But for example, if you look at the teasers that the journals are tweeting, not the authors, but the journals, sometimes it's a bit worrying, because you think, well, in any case, it's a social network, it's Twitter, so people know that it's not exactly the truth. So I can post anything, and no, you cannot, because people read this, and they get the impact of the reading.
Yeah.
So,
and I feel that sometimes just to make it more catchy, people supply a false information, and it's just different from the paper. It's just super different from the paper. When I go back and read the whole paper, it is a different paper than what was.
Absolutely.
Yeah,
you are perfectly right, Benita. And in fact, if you follow the trend, you see that you have a paper that is extremely precise and well balanced with a good title, and then there is the first phase where the social medias will smoothen a little bit, the stuff, and then it will be commented by a second phase where it will be even more smoothened, and 5 steps later you have a different message. On a more on a more general issue, I think social networks are very ambiguous. Because on the one hand, it connects people in a way that has never been before. On the other hand, it disconnects people, and people are totally alone.
Now I see more people communicating science and in more different ways, like SciArt has come to a very strong platform, or science comics had came to a very strong platform. What do you think? Like, since I had always felt that we work from taxpayers' money and they should know what science we do. So what do you think about science communication?
You mean on the open science problem, or yeah. No, but I for the past 10 years, we publish in open access in my laboratory, in full open access. However, that creates some problems which we need to address. To say, oh, I'm in favor of open science publication, but not addressing the problems that it creates is not a good position. So I think we should say, yes, of course, we should all end up going into an open platform and so on. But then what do we do? How do we evaluate people? How do we make sure that the right person, that there's no inequality with people coming from different countries with perhaps less access to platforms or things like this? How can we maintain a good level of quality in research, in academia, by using a fully open platform for publication? This is a very interesting question. There are solutions, but for the moment, the solutions are complex. Okay, because when you ask PIs who are actually quite busy to evaluate someone for a promotion or something, and instead of watching the CV or sort of 5 or 6 pages of statements or scientific statements and so on, you need to go in detail through 10 publications. You say, well, I'm sorry, guys, but I have little time, and it makes the entire system at risk, because you go down with the evaluation. It's like for evaluating papers. You go down and down and down because you say, oh no, sorry, I have no time. And finally, you hit referees who are not used to do this, who are very young and starting. And so I think we should try and find a solution where, of course, taxpayer money has to be transformed into totally open science. This is my position. But on the other hand, we have to think of a system
of the quality check
where we can evaluate people. Because if we cannot evaluate people, then I'm not against the idea of not evaluating people, sort of a complete equality, but how do you run the system then? It's so complicated, But also what I just told you is not totally honest, because if there is such a case where there's a fantastic piece of work produced in the lab, what am I going to think? Let's send it to Nature. Why? Because there's a history of this journal, because this is the journal where things were published, because you feel you're part of a sort of historical movement that okay. So this has to change, but it's deep inside, for at least for people from my generation. It's very deep inside, it's a feeling part of a movement that has built a scientific corpus step by step, with the discovery of the DNA and the structure of the DNA and so on, and then we say, well, okay, if I publish it in an open platform, it will have the same impact. Yes, probably. But we have to change our mindset. That's the second thing. We have to change structures, and we have to change the mindset. And this will be easier for you guys than for us.
This interview is, I think, like so many points to note that what we should be doing in our generation.
Absolutely.
And I think it's an opportunity and a responsibility both in the same time to make it better.
So our next question is specifically for students who are in their undergrad or they're planning to apply for the PhD. So how should they prepare themselves going in the next phase of their life? What are the advices that you would have for such students who are interested in science and taking their major step into the journey?
Well, look, there's two things. First, advice. It's not a word that I like a lot, I must say. And, you know, when people ask me about an advice, young people, I say the only good advice I can tell you is to never follow any advice that old people are giving to you. Okay? Because you have the energy, you have the potential, and people who would say, you know, in my time, I've done this, I've done that. I mean, it's quite useless, in fact. How I do recruit people, this is a good point. So first, I never recruit people without talking to them. I never recruit people without having them talking to everyone in my laboratory in my absence. So whenever someone comes to visit, I talk to the person and then I give the person to the lab, and then I have a discussion with my crew, with my colleagues after, because I think it's not the laboratory is not mine. You know, the laboratory is made by people working in the laboratory. I spend little time in my laboratory, so you can see, you know, someone who is just, you know, you hire someone and you crash the person into an environment without, so it can't be like this. But you know, I'm totally unable to tell you which criteria I would use to hire someone. It is sort of a feeling, you know, and I would say it's, you know, I don't know if you have read this book, which was called Blink by an American journalist called Caldwell, I think, I'm not so sure, but who explains, you know, that in fact the decisions are taken very, very rapidly in a few seconds before you start thinking of the problem, the important decisions. All the rest is sort of trying to convince you that the decision is good or wrong, or say, but the decision, you take it on the spot. So you open the door of your office, you see this candidate, pre-doc, postdoc, and in a way, like animals meeting in the jungle, you know, you say, hey, that's going to work, or that's not going to work. And after you sort of build all kind of, you know, you build all kind of reasons and parameters and classifications and so on. But on the back of yourself, you know that the decision is taken. And often, I must say, often when things did not work really well, it wasn't often the case in the lab. And when things didn't work very well, I had to confess to myself that I knew it would not work very well. You know, it's you know, again, it's human being. You don't go to the supermarket and select the cheese and the piece of meat. You know, you deal with people, and if you want someone working well, you need this person to be happy. That's the only solution. Is the only solution. Okay, you need this person to be in a nice environment that her or his brain is not occupied by conflicts, by problem, by stress, by, you know, anxiety and so on. If you can reach this, it's very difficult. But one easy solution is to try to have a good atmosphere, no pressure, and people are feeling happy. Doesn't mean it's going to work, but I think it's ad minima condition to hope that it will work.
So a few decades back, you were being mentored, and now you have mentored so many people. So what is your mantra to mentorship?
Look, think of what I told you before. Advice it has to be taken for what they are. I would say probably, I mean, that's my view of it, but at least that's what I did. A good mentor is probably you spend as much time talking about anything else but science, in fact. And I would say another point is to be accessible. It doesn't mean to be in the lab and to say, if you want to see me, you know, come and talk to me. This is not what I mean accessible. Accessible, it means to create an atmosphere where people do not hesitate to come to you. They do not think, oh, but you know, is it so important that I should talk to Denis? Or I'm afraid it's going to work and he will. Okay. So you need to create this atmosphere that whenever you want to talk to your PI, you go to your PI.
Yeah.
Not easy because of course there's always a hierarchy, especially for pre-doc, who are joining the lab and they see the professor, as you said, who is a member of the Royal Society and the academy and so on. And so they see someone who will, and you need to deconstruct this. It's not complicated. Huh? You need to deconstruct this because that's the only way to have not to lose time. If it takes 6 months to solve a problem because you can't talk, it doesn't work. You can't do this. So I think that this is really the main, to me, this is the main thing, is to create an atmosphere where people can talk. I would not go to them. Yes, of course I go to them, but I would, I think I would rather let people develop, but be available if there's a problem. Yeah.
Yeah, I think like being accessible and available, like I don't know if it's everywhere, but in a lot of labs in India, it's like the supervisor is the king of the lab, and like they're not very approachable. But
Look. I'm, I always had a problem with this. It's true. I totally understand what you say, and I tell you, I've seen many, and I still see so many.
yeah,
But the problem is mixed. It's like in a family. Until a certain age, you teach your children, and after a certain age, the children are teaching you. And sometimes this second stage doesn't happen in the laboratories. Okay.
yeah,
But people should realize that the people who have the power in the laboratories is not the PI, it's people working, producing goods, producing, results, data. And,
absolutely.
if there's two solutions, the PI is smart, intelligent, and if people go to him and say, look, we would like you to behave differently in your lab to, because we would need something different. If the PI tells you, well, guys, I think you're right. Let's sit down and see how we can do it better. It means he's intelligent. She's intelligent. That's fine. If the person doesn't do this, then the question is to you: what are you doing in this lab? I mean, do you enjoy being in a lab where the PI is nuts? I mean, that's your problem. It becomes your problem. Okay.
Yeah,
So I totally agree. Many persons are abusive in many respects. But on the other hand, the laboratory should not hesitate to tell it. There's no danger in saying this. People don't talk enough on these issues.
So our next question is, what are the new exciting areas and the relatively unexplored areas that you see in developmental biology and which excite you?
You mean if I would start again and look at the question, for example, of timing, how biological systems measure time, it's how time is encoded in DNA, and scaling, how is it possible that look at your arms. How is it possible that you reach the same length, almost to the millimeter? It's nonsense, totally nonsense. Who controls this? Robustness in development. What is the quality control? The equivalent of the quality control. It's crazy. I mean, it's just crazy, And then all the rest. I mean, single molecule stuff, quantities. For 40 years, we've discussed about qualities, and now we are moving into quantities. It's crazy. It's a different world. We're no longer going to say a gene is expressed or not expressed because it doesn't count. But how much? Is it 10, 100, 150, 200, 500,000? And what is the difference? Some colleagues who say that everything has been done in developmental biology, and it's totally crazy. I mean, we are just at the 1%, and the horizon is totally open.
That is so
And
true.
on the other hand, is it developmental biology? Is it a field? Is it a discipline? I don't know, because the success of developmental biology is precisely that it went into so many different fields that it became a context. It's no
Absolutely.
longer a discipline. It became a context to think science, to think life, to think the position of human in nature. Okay, so developmental biology has no sense. Everything is developmental biology. What can you tell me one thing in life science that is not developmental biology? Aging is development. Neurobiology, it's
Definitely.
all set during development. So no, it's very rich. I'm very optimistic.
So yeah, now coming back to our questions, are there any common misconceptions about scientists that you come across?
Well, I think if I had one to mention, sometimes people from the civil society, because sometimes I go giving lecture in in communication of science and so on. Sometimes I notice that people think scientists are smarter than other people in the society. And I can tell you, this is a misconception. That is, scientists are it's a subset of the society, but it's not a selected subset. And it's very fortunate. It's not actually. It is perhaps the only factor that selects the subset is perhaps curiosity. This is, one thing that a trait that I think is quite recurrent, in fact, curiosity. But, it's important because I often hear people saying, am I able to do this? We know, will I succeed? I will never be able to, etc. And, people have to realize, yes, everyone is able. If you have good processes, if you have good skills, experimental skill, and if you are surrounded by good people. And it's not a matter of your IQ and knowing, that you don't have this. All of this is crap. All of this is, science is a job. You need to do it properly, and you need to invest time and effort. But you can be a fantastic scientist by, not being the smartest people in the world. That's a misconception.
What do you do when you are taking break?
I don't. No, look, whenever I can, I go to the mountains. I'm a Swiss born. I go to the mountains, and but if you go high in the mountains, if you hike, you start secreting endorphins, and what do you do with this? Well, you think. And what are you thinking about? You think about science, and constantly. I usually go alone, and constantly. I remember that when you are there, you solve all the problems, even the things which look like the most impossible thing to do. It's easy. Of course, when you're back to the lab, everything has disappeared. It's still very complicated and so on. But it brings you into a sort of a mood where you think everything is possible. And this is a very general feeling. You can ask many people on these, people biking, for example, or running,
Absolutely.
running marathon, after a few I've never done that, but after a few kilometers, you start entering into a sort of a mode where things become easy. Problem tends to settle, and this problem I have with this colleague, easy. I'm going to tell him or her this. Oh, this bloody editor who asked me to revise. Oh, very easy. I'm going to do this. So it's very cool. Of course, after you lose the endorphin and you hit the reality, which is different. But whenever I have time, I go to the mountains.
That's awesome. The mountains call you.
So coming to our last question of today, what do you think about Kitchen Lab?
Great. I tell you, I think it's great, but again, I think it's it, to me, it illustrates that you are simply that you simply are curious, and no one gave you the possibility to exert your curiosity. That's what it means to me. Um, I remember when I was a kid, I would find hedgehog splashed on the road by a car, and then I would go with pieces of wood to dissect and get the intestine out and see what was inside and so on. And if people do not give children with this possibility, they end up doing kitchen lab. I think it's a fantastic thing. I think it's a fantastic thing.
That's very beautiful.
If you could even make a sort of a network of kitchen labs and so on, because it's close to what we call here, the citizen science. When you try to involve people from the society to help scientists in open space, it's extremely important for many reasons. First, because these people are taxpayers. They are taxpayers. So, the more you involve them, the better it is for them to understand what is the aim of science. It's not so easy for people outside to understand what is the aim of science. And I think if you invite people in your kitchen and they start to see all these things and so on, they may sort of understand what is an experiment. Very difficult to understand for someone outside. What is a biological experiment? What is it? What do you mean by this? For you, for me, it's obvious, but not for everyone.
Yeah.
So I think it's great. I'm very admirative of this. You see,
So much.
in Geneva, people in the 18th century, 19th century, that's what they were doing. They had they were doing science home because there were no laboratories, so they were cutting things in pieces, looking at the pond for frog embryos and so on. Go ahead. Don't give it up once you will have your own laboratory. Or you can do the opposite. When you will have your lab, you make a kitchen in your lab.
Thanks a lot for today. And
Benita, Schumann, that was a great pleasure.